Dr. McNeil, Alex Shaw behind the camera. Termination transcription in eukaryotes. So, you guys should know how termination transcription happens in prokaryotes, in either a row dependent, where row, a helicase slides down an mRNA, knocks RNA polymerase off the DNA method, or the row independent way, where a stem loop forms and that stem loop binds to a protein called NUS-A on the bacterial RNA pole, and that pulls the RNA pole right off of the DNA. Okay? But this is eukaryotes. This is us. And we don't do it like that. We don't do it like that at all. We use a totally different system. So I'm going to orient you here. Here is a eukaryotic gene. Not all the parts of a gene are drawn here. We've got a +1 site, transcription start site. It's on the coding strand. You have 3 exons here, exon 1, exon 2, and exon 3. Those are protein coding parts of the gene that will ultimately be part of the final mRNA. We have intron 1 and intron 2. Intron 1 down here has been spliced out. There's no more intron 1. Intron 2 has yet to be spliced out, and that will occur in the future. So we have 5' UTR exons. That intron needs to be spliced out. Exon 3, and our 3' untranslated region. So how does this work? Well, here we have RNA polymerase 2. RNA pol 2 in eukaryotes is the major workhorse that's the RNA polymerase. It writes essentially all of your messenger RNA. Why is that important? Well, messenger RNA needs to be polyadenylated. And so the major thing we're going to do here is place a poly-A tail on our developing, maybe what we could call our maturing mRNA. Okay? And once it's done maturing, it'll have 3 things. It'll have 1, a 5' methylguanosine cap. It will have all the introns spliced out. And 3, what we're focusing on here, the poly-A tail. Why do we need a poly-A tail? Well, a poly-A tail in transcription in eukaryotes essentially protects the 3' end of mRNAs from degradation. Why does it do that? Well, mRNAs look a lot like a viral genome. A lot like it's just a naked, one single-stranded mRNA floating around that looks a lot like viral genomes look. So we want to make sure that our mRNAs are protected from degradation. We're going to do that by jacking 200, 300 A residues onto the end of it. How does this work? Well, RNA polymerase is bound to the DNA. It's been transcribing, writing in a 5' to 3' direction, utilizing the template strand as a template for synthesis. What do we get? We end up here with RNA polymerase rolling down the 3' UTR, and there's no real stop inside, right? There's no real signal. But there is a stop signal, and that signal in the mRNA looks like this: AAU, AAA. And that is what you would call the poly-A site. And it's a signal to RNA polymerase to stop. Well, how is it the signal to stop? I've already drawn RNA polymerase going past it, so either I'm stupid or there's something else involved, right? But both of those things are true. I am stupid, but there is something else involved And what else is involved is that RNA pol is, well, it's about 20 different proteins. Okay? So the holoenzyme is it's a huge multi-protein complex rolling down its DNA. Remember, it's not on there super strongly, but it's on there well enough that it can chug through several million bases, because a lot of human genes are multi-million bases. Okay? But it hits a poly-A site. What happens at the poly-A site? Well, one of these 20 proteins in the RNA polymerase is called CPSF. That stands for cleavage, promoting, specificity, factor. So we're going to promote cleavage of the mRNA in a specific place. Okay? So CPSF is a protein. And it's bound to RNA pol 2. So what's awesome about CPSF is that it's been bound to RNA pol 2 ever since RNA pol 2 started transcription. So it's just hanging out. I'm going to draw like a little tail out behind RNA polymerase. This is CPSF. It's been hanging out, riding on RNA polymerase, going down the template strand of DNA, writing the new mRNA, and CPSF is what you can consider to be scanning the mRNA. And when CPSF, when one of its protein subunits identifies AAU, AAA, CPSF, a protein, binds to that mRNA. And because CPSF is both bound to this mRNA and bound to RNA polymerase, it stops mRNA polymerase. So, I'm going to go with one or two steps here. One, CPSF recognizes and binds the poly-A site. And in humans, that's AAU, AAA. 2, that binding stops RNA pol. Okay? But now what we've done is stopped RNA pol. Hasn't really fallen off the DNA yet. Okay? And it's not going to necessarily fall off. It doesn't fall off as easily as RNA polymerase does. So the next couple steps, nobody's really solved yet how exactly the mRNA gets cleaved, but it does. So the mRNA is cleaved and it results in this whole thing with CPSF bound to this mRNA falling off from the DNA, falling away from RNA polymerase, and what we have is CPSF and generally about 30 bases of RNA with it. Okay? Here's what's awesome. All that does is get us to termination of transcription, but it doesn't do what we need to do, which is to put in our poly-A tail, to polyadenylate our mRNA. So CPSF is there. Now I'm going to erase RNA polymerase. RNA polymerase is still hanging around right here. It's bound to CPSF still. So RNA pol is still here, but it's not really doing anything right now. The other interesting thing that's bound to CPSF, I'm going to draw it in a big orange box here, is this thing called PAP. And PAP is the poly-A polymerase. And poly-A polymerase is interesting because its only real function is to write A's, to write adenine on the back of this mRNA. So we have a free 3' OH off the end of this newly formed pre-mRNA. And poly-A polymerase is here. Poly-A polymerase begins writing A's onto the end of that. A. And that keeps happening. The first few A's get written, and that actually increases the strength of the bounding of poly-A polymerase onto the mRNA. But all these things are still physically tied together. CPSF is bound to RNA pol. That's bound to the poly-A polymerase. So I'm going to put here 4 poly-A pol is in position to write poly-A tail. Okay? So poly-A tail begins to write that, and it's shooting a whole bunch of A's out. But remember, CPSF is bound to this AAU-AAA. PAP is bound to CPSF. These things can't get apart from each other. So what begins to happen? Well, poly-A's keep getting written. They write A after A after A after A after A. And so what happens? These A's have to go somewhere, and it begins to branch out and bulge out around here. And poly-A polymerase 5 can write about 250 to 300 A's in a row before it falls off. Why does it fall off? You can only crowd so many A's into this area before it begins to push against CPSF, push against poly-A polymerase, and push against RNA polymerase to cause this whole thing to fall off. So once it's written a whole bunch of A's, this whole complex comes apart, and what do you end up with? You end up with an end of your molecule. I'm just going to draw it from exon 3 over. Exon 3. You got your 3' UTR. You've got your CPSF binding site still existing. Then you've got about 30 nucleotides. Then you've got A's times about 250. Okay? Now, the only reasonable question left is how does this protect it? How does this protect our mRNA? These A's allow it to be safe, because if you imagine a exonuclease, that's an enzyme. Exo means outside. Nuclease, it destroys nucleic acids from the outside. If it tries to chop up this nascent mRNA from 3' end, it's got to churn through 250 A's that are meaningless before it begins to get into the regular part of the mRNA. And it still has to chop through this whole 3' UTR before it actually begins to destroy the protein coding part of the gene, of the mRNA. So what do we actually end up with? We end up with a lot of buffer room that the poly-A tail creates on the 3' end of a pre-mRNA. Termination transcription in eukaryotes.